at segregation of alleles. So let's say you have a cell that is on two chromosomes. One chromosome, you have the big A allele, and the other chromosome, you have the little a allele. These are allelic pairs. So again, you have one chromosome, one chromatid, one double-stranded DNA molecule. Then you have replication, and you're going to have two chromatids held together by the centromere. This is one chromosome, just as this is one chromosome. These are not two chromosomes. This is one chromosome, one chromosome. So two chromatids held together by the centromere and cohesins. Then they align at the metaphase plate like this. And then again, you have one bivalent, consists of two replicated chromosomes, one red, one green, four chromatids, two red, two green. And during the first meiotic division, then the homologs separate, so that having the big A allele goes to this pole, and the one having the little A allele goes to the opposite pole. During the second meiotic division, then you have separation of the chromatids and the centromere, and then you end up with two gametes or cells, haploid cells that have the big A allele, and two that have the little A allele. So you have basically two big A's to two little A's, or 1 to 1. And this demonstrates Mendel's first law, the segregation of alleles at a locus. Okay? That's in plain words. This is Mendel's first law. Are there any questions thus far? Okay, so let's okay, so now let's look at a cell that has 4 different chromosomes, okay, and these chromosomes, so these red and green are a homologous pair because they carry the big A and the little A allele, so this is one set of homologs. I put it in different colors just to illustrate this, but in actuality probably should be in the same color. And this one carries the big B, and the little b allele is a different homologous pair. Okay, so the number of chromosomes, so this is a diploid cell with 4 chromosomes, and it has big A, so the genotype is big A, little a, big B, little b. The slash here means it's a chromosome, so the one resides in one is allelic. Big A is allelic to little a, big B is allelic to little b. Okay, then we have replication. Okay, so we have 4 replicated chromosomes. So, and 4 replicated chromosomes, and 1, 2, 3, 4, 5, 6, 7, 8 chromatids here. Okay, so now we have the alignment of the homologs at the metaphase plates. I told you that red and green are one set of homologs, so they align like this. Purple and orange is another set of homologs, so this is a different set. So different pairs of homologs align with each other. So if you have 10 different homologs, and I don't put 10 here because I'll run out of space, they will be aligning on the metaphase like that, each with this homolog. Okay, so here we have then So this is the alignment at the metaphase plate. You have two bivalents. This is one bivalent, and this is the other one. And you have, in total, you have 8 chromatids, but each bivalent consists of 2 chromosomes, 4 chromatids. 2 chromosomes, 4 chromatids, but total number of chromatids is 8. Okay, so now we have the first meiotic division where the homologs will go to opposite poles. So you have red and purple go here to this pole, and green and orange go to this pole. Okay, so you have the one carrying the big A allele and the big B allele at this pole, the one with the little a and the little b allele on this other pole. Now you have the second meiotic division where you have the separation of the chromatids going to opposite poles, and you end up with 4 haploid cells. 2 of them carry big A, a red and a purple chromosome. So they have big A, big B alleles. The other 2 carry a green and an orange chromosome with little a, little b. So you have 4 meiotic products, and you have 2 big A, little b, 2 big A, big B, and 2 little a, little b. Okay, so notice one thing. Here I align the chromosomes. Let's see if I can. So I cannot pull the annotation tool, doesn't show up. Okay. Notice that I have aligned the chromosomes in this fashion. Red on top, green here on bottom, purple on top, orange on bottom. But you have there is an equal probability that the orange will be on top and the purple will be on the bottom. There's 50% chance, and it does occur. And let's see what happens when you do that alignment. So remember here I have red, purple, green, orange on the bottom, right? And then the ones on top. Now let's see what happens when you have the other arrangement. Okay, so now chromosomes, the same chromosomes, replication, the homologs align at the metaphase plate, but now the so red aligns with green, orange with purple, but I switch the position. So before I had purple on top and orange at the bottom, and it does make a difference in the product that you get. So you have the first meiotic division. Now you have red and orange, the homologs. So first of all, let me backtrack. This pair, the big A, little a, homologous pair, separate from each other. So you have red going to one pole, green to the other. Then now this homologous pair, orange, purple, separate with orange going to this pole and purple going to the opposite pole, such that now this cell has red chromosome and an orange chromosome, and this one has a green chromosome and a purple chromosome. And now we have a second meiotic division. Okay, and we have that the chromatids separate, and you end up again, every case, 4 haploid meiotic products, where you have big A, so you have 2 of the cells will have a red and an orange chromosome, and 2 of the other cells will have a green and a purple chromosome. And the alleles here will be big A, little b, big A, little b, and little a, big B, little a, big B. these are not allelic to each other. Yes, they are not allelic. That's why I put them in different letters. So the four meiotic products that you get are 2 big A, little b, to 2 little a, big B. Before we got 2 big A, big B to 2 little a, big B. So the arrangement of the homologue at the metaphase plate results in shuffling of this information, okay? How they, who goes with whom in the final product. Okay, these two slides that I've just shown demonstrate also Mendel's second law, the law of independent segregation. That means that alleles that reside in, or genes that reside on different chromosomes segregate independently of genes that reside on other chromosomes. In other words, the A pair segregates independently from the B pair. Okay, so now let's, I said that we have recombination during this stage. Let's see what happens if there is recombination. Are there any questions thus far?
No, not for me.
Okay. Okay, so now